Okay. So, good afternoon, everyone. It's my great pleasure to present you today, announce you the plenary of Jeppe, who has completed, after completing a master and a PhD in the University of Southern Denmark, has completed a postdoc in Germany, Texas, California, Norway as well, and is currently a postdoc in the University of Southern Denmark, and he will now present you your oh, sorry, University of Copenhagen now, my bad. And he will now give you an overview of his rich experience with bioacoustic during his career. Jeppe?
Thank you so much. Yeah, I know it's a little bit confusing because it says University of Southern Denmark here, but that's just a little bit of the history about me, because I grew up in a small town called Middelfart, and in Danish it's not funny at all. I know it is in English. But this is Middelfart, and it looks very pretty there. But the thing is that in the waters around Middelfart, we've got the highest population density of harbour porpoises anywhere in the world. And I quickly, as a kid, found out that these harbour porpoises see their world, their murky, dark world, by using sound. And my mind was just blown. How can that happen? Of course, we are talking about echolocalization, right? But then I also realized that they receive the sound via their lower jaw, that then let the sound up to their ear, and it's just like, wow, that's so cool. And that's sort of like the beginning of my path to being a bioacoustician. Always been a geek, still, and always will. So, as Patrick said very correctly, I got my PhD at the University of Southern Denmark, where I worked with sound production in birds. We're going to get into that later. But during my studies, I developed a little bit of love for wolves. I think they're super interesting animals. So I actually found a temporary position in Minnesota where I captured wolves and put on radio collars and tracked them from a small plane. And I had so much fun over there that shortly after coming home, another position in Tasmania, very far away from Minnesota, where I was working with Tasmanian devils, whereas wolves are super soft and cuddly and basically teddy bears with teeth. Tasmanian devils are smelly, aggressive little bastards that you should stay away from. Yeah, I'm very objective in my description of animals. So, there's a lot of brilliant scientists that spend their entire life and maybe even get a Nobel Prize by studying something very particular, like one small protein pump in a cell membrane or some specific species or something. That's not me. And I'm never gonna get a Nobel Prize. I'm all over the place. So it's like I've been working with a lot of different species right now, and that's basically why I also had the title for this talk. Is like bioacoustics can lead you to do a lot of weird stuff with different species. That sounded wrong. So as you know, I've been working with Tasmanian devils. I've been working with goats. I've been working with cod in Norway and the harbour porpoises. Also like a little bit on the sideline at SDU, the wolves, and I've done some software to analyze acoustic behavior in dolphins. And despite being not focused at all, messy, might some say, I've still been able to get into some fairly good journals. So maybe it's not that bad an idea. But I'm going to focus on a few species today because I only got 1 hour to talk about. So I'm going to tell you a little about sound production in birds because that's where I started with. I'm going to talk about blue whales and fin whales. I'm going to talk about Hawaiian monk seals, and finally, of all the weird animals, pigs in the agricultural industry. But let's start where it all started, with sound production in birds. And in order to understand, get us into the mood of sound production, I've got this video of human sound production. Now we are coming down to epiglottis and down to the vocal folds. That looks like something that makes this video age restricted in Denmark. You get the idea. So is this working? Good. So what's actually happening is that what you look down at was looking down here, right? With the epiglottis, that was the white one that you could see. That's sort of like a lid that goes in and closes off your windpipe when you're eating, which not always working. But beneath that, we've got the vocal folds, as you saw in the video. And when we are vocalizing, then they are close to each other and they're vibrating. And the vibration of these vocal folds is what creates our voices. So it's pretty much like a grand piano. So the longer the vocal folds are, the lower the frequency. So like if you played a tone here, there would be like boom. That's not how a piano sounds. And over up here it's like ding, right? That the longer, the deeper. We also notice from humans that a lot of male got an Adam's apple, and that's because the vocal folds are longer, so they basically stick out of the neck. And we've got men got about 100 hertz fundamental frequency of the vocal folds, whereas women is about 140 to 150 hertz. And there's of course some statistic uncertainty here. But also the weight of the vocal folds means something, that the heavier the vocal folds are, that also lowers the fundamental frequency. In this company, there's no reason to explain fundamental frequency right now. So that's the two. Well, there's a third and very important one, the tension of the folds. The more tension there is on the vocal folds, the higher the fundamental frequency is. And that's how we adjust it. And apparently it's something that you have to practice. I haven't sung for like 20 years, and not that long ago my niece then asked me to sing a lullaby for her. It sounded horrible. So please don't ask me to sing anything anyway. But what creates these vibrations that we talked about that we could not see in the video? We all agree that the vocal folds oscillate, right, and collide. That's how we produce sound. Is it muscles that collide them? No, it's actually not. Because if you look up here, that's the contraction time of a normal muscle. And it takes like 100 milliseconds. That's like 0.1 second, right? That means like, and this is even a fast muscle then, that we could never produce a sound that's over 600 hertz, right? Because 100 milliseconds, that's 10 oscillations per second times 60, 600. So what actually drives this vibration of the vocal folds is the passage of respiratory air. This is an illustration I made that's in a textbook that's coming out very shortly. Not doing any advertising here, but just like exploring animal behavior through sound, volume 2 from Springer. There's like this cycle of the vocal folds that if we started here where it's fully closed, and this is towards the head and this is towards the lungs, then in sound production, the first thing that happens is that they start opening. And you might see that the shape of the vocal folds is not the same shape as up here. There's much larger distance towards the lungs than there is towards the head. That creates a overpressure, symbolized by the blue color. And this overpressure inside here helps open up the vocal folds. So it's pretty much like when there's heavy traffic on the Autobahn, just been driving through Germany, that there's more air in there because it's a convergent shape. So like if there's a crash here, then it has to go around all of the cars, so it get closer together. That increases the pressure. So that pressure then helps open up the vocal folds. And then we get to a fully open situation of the vocal folds, and then we get to the closing phase where we have what's called a divergent shape. And a divergent shape just means that the area up here is much larger than the area here. And that's the case that we see with planes when they're taking off. That on top of the wing, there's a under pressure. And that sort of pulls the plane upwards. And that's the same in our vocal folds, that this pulls in the vocal folds and help closing. And that's something called the myoaerodynamical theory. And the aerodynamical comes from what we've just shown here with the pressure, overpressure and underpressure. And the myoelastic part of this, the name, is that the reason why there is this cycle is that the muscular tissue, this vocal fold tissue, has elasticity in it. So that's a very short explanation of how the vocal folds functions. So to say it in another word, we have two vibrational components. Vibrational component 1 and vibrational component 2. So vibrational component 1 is the one that we all know, that the vocal folds vibrate and collide with each other. So they're open and they're closed. And what I've just explained now is the vibrational component 2, that they can be a divergent shape and a convergent shape. In order for any sound production to be named meet the myoelastical aerodynamical theory, these two components has to be present. We know what drives this sound production. We've long suspected that this is the case in birds, even though when you listen to a bird, it sounds like it's a whistle instead, right? So no one knew for sure that this was actually the mechanism behind it. So we wanted to do that. The case in birds is that songbirds to the left got two independent sound generators. So it's like if you had two larynxes with each their voice actually tuned to two different frequencies in birds that you could use simultaneously, that you could control individually. That's pretty cool, I think. Whereas in non-songbirds, like a pigeon that I've been working on a lot, and that's over here, that's why they call me Pigeon Boy back at SDU, there's only one sound generator. And this is sort of like the vocal folds that we see here. They are called the LVMs in birds. And here there's lateral medial labia and lateral labia. That's sort of like the vocal folds in birds. So for birds to make sound, it actually only takes two things. The song organ of birds are sitting within a pressurized compartment of the respiratory system called the interclavicular air sac, ICAS for short. And here we've got the vocal folds, as we call them, the LVMs. And there is no muscles needed for vocalizing in birds. You can cut the nerves leading to the syrinx and it can still make sound. Actually, a lot of birds will constantly make sounds if you cut the nerves, because birds are made to make sound. They're not made to be silent like we are. So all it takes is that it's the correct air pressure in the ICAS and the correct pressure coming from the lungs. Then it will vocalize. We use this for our PhD thesis to do some experiments where we created, well, mostly my supervisor, to be honest, created this chamber where you could then dissect a syrinx, a song organ out of different kind of birds. This box we made in different sizes because we also put a syrinx of ostrich in there. That's a big one. And zebra fins, that's super small, as you know. And then we can just simulate that the syrinx, the song organ, is still within the bird. So we can control the pressure outside and the pressure inside of the syrinx. And then we can see what's happening. And we did that. And this is what we saw. This is a pigeon syrinx. And if we shine light for it, we can actually see the labia inside and we can then draw it. You want to see it again? So if you looked at the red and green line, then you'll see that as is actually a closing cycle where it's divergent. That there's more space up here than there is down here, right? And we see that during opening, it is actually converging. There's less space up here than there is down here. So we've got the two vibrational components. We see that they collide, that they vibrate out and in, and we see this asymmetry of the shape of the vocal folds. So we proved that birds use the same physical mechanism as all mammals do for sound production. And that, of course, have big consequences for using birds even more as a model species for human vocalizations. Most parents tend to get slightly angry if you ask them if you can put some electrodes inside the larynx of their baby, whereas it's a bit easier to get permission from zebra finches. So we then continued this because this was pretty cool. So I went to Maine and worked with an engineering group over there because we wanted to see if we could then model the airflow and the kinetics of sound production. This is still a pigeon that we're looking at here. Because if we could that, we could maybe also do it for humans, because each year 3,000 people lose their voice when they're getting surgery in their larynx. If we could do a model that this surgeon then could see, like, okay, if we move this tissue, will it still oscillate? Will the patient still have a voice afterwards? Maybe we could do something really good for a lot of people around the globe. And they're actually starting to work with this medically. Of course, it takes some years before we are doing the first tiny little step until the doctors know how to do it correct way. I had some really nice... Something froze now. The previous one, maybe? Had some pretty cool animations here. Yeah, you've seen this animation before, right? That we can see how they move. And then if we look at the next Can we start the next video? Then we see it looks very similar, right? And if we, can I go further? Yeah. We see that the fundamental frequency of the simulated vocal folds and the real vocal folds are also very close to each other, like 150 to 140 hertz. That's pretty close. Even the sound pressure that we get after of the simulated syrinx of a pigeon is pretty similar to what we saw in real life recordings. And doing some statistics, we can do some cool stuff about seeing what is the opening between the labia in both the simulated and in the original one. And we can see pretty similar patterns here, right? That it's sort of like, you see the blue color is when they are closing down to zero, so they are colliding. And we see, just like we expected from a meat, that they collide at the bottom first and then they collide upwards as they move. And we see that in both cases. So that's a PNAS paper there, right? So that's pretty nice, and a lot of the parameters also show no significant difference between the simulation and real life. So I think we've got a fairly good model. Okay, enough about birds so far. The next thing I want to talk about is that I got a position in the outskirts of Houston, and me being a space geek, that was super exciting. I could come and visit NASA headquarters there. And it was in a small town called Galveston on an island with a 30-kilometer long beach, awesome Mexican food, good margaritas, nice people, because we wanted to see if we could find an automatic way of detecting baleen whale social calls. But why? Well, let's start out with a bit of history first. Around the globe, there's these harps submerged in the oceans, and it's basically a listening station with a release mechanism so that you can go out and you can send a signal down to it, and then it releases and goes up to the surface, and you can pick it up and retrieve the recordings that you've got. It's basically just like a sound trap, giant sound trap. So it's the hydrophone and batteries and computer and storage device. And this is the ship that we used in Texas, and this is what you see to the left here is laid on the deck of the ship before we are going out and submerging it. They were actually put in the oceans by the US Navy to listen for Russian submarines. There's quite a lot of years now where we did not have to be concerned about the Russians. Have changed slightly right now, but all these years we could just use them for listening for something useful instead. If you ask me, whales are way more interesting. So why do we want to detect these baleen whale social calls? First of all, there is no automatic detector as it was before we started this, because they are more variable. So you cannot use traditional methods like spectrogram correlation, match filtering, stuff like that. So that makes it interesting, right? That we also want to detect these types of calls. Also because these variable calls are also emitted by the females. All the automatic detectors that was out there could only detect songs that's very stereotypical, and songs are only emitted by males. So we had no idea if there's a big difference in the migration routes and the population trends between males and females. So I think it was pretty essential that we tried to develop this detector. So because of the variability, I got the idea, all by myself, that maybe deep learning could be a good idea. Everyone here, of course, knows that a deep learning network is really good at classifying images. So it can quite quickly say, this is a cat. And this is back in 2019, so that's like Stone Age when we are talking about AI, right? What we thought was that spectrograms are also images. So could we train a neural network to say that this is a D-call, a social call from, and this is how it sounds. Can you hear it? deep sound. 2 more coming. And now this one comes. That's pretty good sound. I also had a sped up version if the speakers could not handle those low frequencies. Just increase the sampling frequency with 5. So, what you need to do when you're using a neural network is you need to train it, right? And often it's a question about showing it a lot of examples of whatever it has to learn. So in this case, where we have to train it to recognize between a cat and a dog, we just show it. That's me teaching, by the way. You see the hairdo is perfectly fine, right? So that's how we train it, and that's also what I did with the whales. That there's a data set out there called the DCLDE 2015 data set from California. Someone's nodding their heads that know it. With annotated data set from outside California that you could use for training neural networks like this. So what I ended up with was that I actually used 2 networks. I first used a region-based CNN, and that's a network that's specialized in finding where in the image is what we want to find. As I said, this is not just a bad excuse, but this was pretty much Stone Age when we are talking about this kind of networks. So you could not get any pre-trained networks that's region-based. So I had to create my own network and train it all from the bottom. So the accuracy was not that good, but that did not matter because I just wanted to detect all regions where there could be a call. And then afterwards I could use a CNN, a pre-trained CNN, ResNet 18. That's, I cannot see where he's sitting. Someone talked about ResNet 18 earlier today, using that. And then we could get some pretty good data out of it and can tell us there was a blue whale there was a fin whale there at that time, and there's like 78% chance that it was a whale. Because our data set for whales are huge, I did some small modifications to increase the speed of this network. So first of all, as I said, I had a custom-made region-based CNN that was fairly shallow, so it had a lot of false positives, but that did not matter because we had the CNN afterwards. But also the anchor boxes, that's the regions that it's looking for. Where is what we're looking for? If you're looking at an image of sailboats, then these sailboats can be at different distances from the camera, right? If it's close, then it's pretty big. It's further away, then it's a bit smaller, and if it's really far away, it's really small. And the neural networks that's been trained for this kind of images need to be able to find all these different sizes of anchor boxes. That's not the case with a spectrogram. The calls are always at the same distance from the camera, right? That it's pretty much same size. Of course, there's some variation, but it's not nearly as we see with the sailboats. So you can go in and then you can ask the neural network to, you don't need to look at all these different kind of sizes. You just need to look for kind of this size. And that also increases the accuracy and it increases the speed. And then finally, got this animation that they're the same size. And finally, as I said, use ResNet 18 instead of ResNet 50, also because of the speed that the ResNet 18 was much faster than ResNet 50, and we saw only slight decrease in accuracy of the two networks. So we ended up with 2 detectors. Of course, these should go all the way up here, be located up here. That's not the case, but it's still the first practical detector for detecting the social calls from blue whales and fin whales. And Scripps is, I'm working together with Scripps still on like moving forward and improving the situation all the time. I've been doing workshops with Scripps, and whoever is into marine mammals know that I was like It's basically Cambridge for marine mammals, right? So I think that was pretty cool. And third animal. Yeah, I've got an entire hour. I need to do some different species here. It's the Hawaiian monk seal. It's highly endangered. And I'm working together with University of California in Santa Cruz, where I'm also a research associate. And for these Hawaiian monk seals, we would like to create a detector, and we want to investigate the call types that they make. And we also want to look into, that was mostly me, investigate the syntax of the vocalizations. In 2021, Gillian Sells, I don't know how many of you know her, but together with a lot of brilliant people and with Lene Reismuth, like one of the giants in bioacoustics, made this paper about that Hawaiian monk seals make underwater vocalizations. And they found 6 different call types that they produce, and they recorded it from 2 individuals, Hoak and Koa, and that they also found that 90% of these calls are produced within a bout, that they make different calls close after each other. So it's like basically a sentence that they are making, right? And there are some examples of the calls here. There's a croak and a groan, growl, moans, rumble, and very short whoop. And this is an example of a sentence that they are making different kind of calls in a row. They manually took 2 days out of each week and then annotated for 1 year, the data from 1 year. But they had 2 years from both animals. So we wanted to see if we could go from those 2 days a week to everything. So basically 4 years of data that we wanted to analyze using the detector. So first of all, we had the annotations from this paper that then I had some students in Norway improve on, and then I made a graphical user interface. I'm a MATLAB geek, so everything I do is in MATLAB. I'm that old, yes. I made this graphical user interface, and then Gillian Sells went through these 4,000 annotations that we had from the published paper, and actually found 1,000 extra calls been missed in the 2 first rounds of going through this data. That's pretty mind-boggling, right? That was it 25% of the data was missed after 3 rounds of going through. So that tells you about manual annotations. So we found these calls and could annotate the start and end of them, because then we've got a really good training set for our net. And for neural network, I chose a YOLO network. The specific about a YOLO network is that, first of all, it's a region-based CNN, so it detects and classifies. And why did I do that? Because 90% of the calls are in sentences. So they're very close to each other, so we need a network that first finds this is where it starts, this is where it ends, so that we do not mix all the different calls together in the sentences. But YOLO also got another cool thing, is that it's got 3 heads, it's called. And these heads determine what kind of anchor box, what size of region of interest that it's good at. So what we did was, because these calls are very different, a growl can last up to 20 seconds, and a whoop takes like 0.5 seconds. So it's very different sizes of anchor boxes needed for these different type of calls, right? So we could say that, okay, the croaks that are sort of like medium, a couple of seconds long, we can set that to 1 head, and then we can set the ground, the really long ones, to the 2nd head, and the super short calls to the 3rd head, and still keep really good accuracy in this way. So after some modifications and a lot of training, and I'm on a Carlsberg Foundation grant, you know, the beers from Denmark, not making Tyson here, but they gave me good money for building my own computer, so I made a really nice computer and got it up and running. And after 8 hours, I think it was, when I finally got the right hyperparameters, we actually got 95% accuracy on 10% of the ones that we saved 10% of the data that the network had not seen. And what we get out is annotations like this. So you can see there's a growl here that the network says, okay, here the growl starts and here it ends. So we could go through all 4 years of data in 13 hours. And it had taken years for that paper from 21 for them to go through the 2 days a week for one animal. But does it match up? Do we see the same on the complete data that they did manually? Because you know you can test a network and it looks like it's working fine, but you're always a little bit hesitant. I also got imposter syndrome. That's like, did I make a mistake somewhere? So we looked at what about the distribution of the call types among these seals. And if you look, for example, at grown, 3%, this is the published data, and this is what my automatic detector found, 19, 21, 16, 18. There's a little bit difference here. But in general, you can see there's like a pretty strong image of that it looks like the same in both of them. And then we also looked at temporal trends. So this is the published data with the 2 days for the same animal, and then just 7 days a week. And you'll see there's still the same trend, but of course this is higher because there's more days now to count. But it all still looks like the AI matches the manual annotations. Then I looked to the syntax. And this is a pretty weird plot. But these spouts, these sentences that the seals can make, can be different length. And we wanted into like these 6 call types that we've got, where are they located within the bout, within the sentence. So I made a plot like this, because then you can go in and say, okay, we can say croak, and then we'll say for a sentence that's like 10 calls long, how is the distribution of the position of croak within that sentence? And that looks like it's pretty much even, right? The croak can be anywhere. Whereas the groan, if you go in and say like, oh, we can take 15, that looks better. Then it's mostly at the very end. If you go down, it's 15. So groan seems to be like a period. It's like when you're on the phone and with your old grandma, it's like, yeah, goodbye. That's it, right? Whereas what I think also is pretty interesting is the moan. That if you look at this structure, it looks like there's sort of like a line going down here with a bit higher color, right? So it looks like it could be a call that's sort of like, once again, when you're talking with your old grandma, it's just like, well, I'll also bet you're trying to end the conversation, but it's not the last word. But it's like a sign that the sentence is ending. And I think that's pretty interesting in a seal. You know, we've not seen this before, that there's definitely some structure to the syntax, right? We also looked into, is there some bias in the transition? So given that the seal emits one call type, is there some bias in what's the next call type gonna be? And it looks like, first of all, when we look at this matrix, it looks pretty symmetrical along this axis, right? So that we see that if the first call is a croak, quite often the next one would be a growl. And if the first one is a growl, quite often the next one would be a croak. And it's very got higher bias here. And it's also the case with moan and rumble. Rumble, moan. I think that's pretty interesting. Our hypothesis is that maybe these calls are related in somehow in some dimensional space, but this is not even published. This is brand new stuff that I'm just showing here. Patrick had the good advice that never talk about something that you're not 100% certain about. I'm not certain about this. Another thing that we also think is pretty interesting is the circadian rhythm, especially with the groan. As I mentioned, we had two animals, right? We have Hoa and Kekoa, and both of them apparently really like to make groan calls in the afternoon. Why? We have no idea. But there's also like, and my dog is dreaming. But also other tendencies like in general, quite often around midday that we see some slacking. Could be something with the feeding, I don't know. But let's go to the last animal because I've got 10 minutes left. Pigs. I know I was part of a documentary called If Pigs Could Talk that's been released across Europe. How many have seen it? Oh, not one hand, come on. Just give me a pity hand, someone. Well, it's been released via Arte in Germany, in Switzerland and France, so you can probably see it online. Because, I'm a bioacoustician and it's not always about marine mammals that I'm working on, but just pigs in general. And we know that pigs in the agricultural industry, there is some animal welfare issues, right? So let's talk a little bit about animal welfare, that it sort of like relies on 3 pillars. So there's the basic physical and health function and functioning, right? That you have to, in order to be happy, you need to be fairly good condition and health, but also the emotional state. If you're living in eternal fear or being stressed by someone higher up in the hierarchy, like we all know from our supervisors, but also an ability to express normal, species-specific normal behavior. You need to be able to do that to be happy. And if all those 3 things are present, then we can talk about good animal welfare. So how can we monitor the mental health, the emotions of the animals? So let's first start with a framework. Elodie Briefer that I'm working with, I think at least one of you, she mentioned, had been a master student with her. Was that you? I was supposed to say Haydn. There's valence, there's negative valence, that's all the unpleasant things that an animal can be subjected to. That's decreasing the fitness of the animal. And that's something that the animal would like to avoid. Whereas we've got positive valence, that's all the things that increase the fitness of the animal, that the animal would really like to get more of, like treats, for example, or belly scratches, butt scratches. But we also got, on the other dimension, arousal that goes from low arousal up to high arousal. That's both for the negative valence and the positive valence, and that is defined as the bodily activation, that how strong is the feeling. So we've got negative and positive, and then how strong the feeling is, this 2-dimensional parameter space. So how are they expressed, these feelings then? Well, we all know about facial expressions. You can probably see on me that I actually like talking about this, despite that it's an entire hour's worth of talking, and enjoy being here in beautiful France. And you can probably also see on my dog quite often that he's pretty relaxed, especially when he's dreaming. And facial expressions and bodily postures. But a lot of studies, as all of you know, no doubt, shows that the vocal expressions of animals can also tell us a lot about the state that the animal is in, right? And some of the most important parameters is the vocalization type, the rate, and the structure of the vocalizations. But especially the frequency means something in many cases. You all know that quite often, not always, far from always, but quite often higher frequency is something associated with negative valence, whereas lower frequency can be something that's associated with positive valence. So in 2021, that's the second time I mentioned that year, we published a paper with classification of pig calls produced from birth to slaughter based on their emotional valence and context of production, and got a lot of media attention on that one as well. You see, I'm far all the way down here. So it's Elodie that I'm still working with today. I was in charge of it. This is a director that's gotten a lot of prizes, a Danish director, Miki Mistrati. And he read this paper and is like, can we use this in real life? And contacted us if we could be interested in being part of a study in real life to see if we can classify the pig calls using AI from real agricultural industry. So what we did, what he did, was that he found 4 conventional farms, and in Denmark, a 100 kilo pig is legally allowed, need at least to have 0.65 square meters. That's nothing for such a big animal, right? Or am I too subjective again? Organic farm, there's 2 major improvements here. First of all, they are born outside. So the mother pig and the piglets are walking around with their nose in the mud in the beginning, and then moved into ordinary stables where they then got 2.4 square meters per pig afterwards. That's a little better. And then we finally found 2 free-range farms where the pigs are living outside their entire life. And in those 3 different categories of pig farms, we then, Miki Mistrati did with advice from us. We did not know anything about which sounds came from which farms, so that it could be completely neutral, what we found out. So it's completely blind, the sounds that we got from them, and could we translate them into animal emotions? Could we find out if the pigs are actually happier in the free range in the conventional farm? You're gonna see the slide tomorrow, some of you as well, slightly modified. But whatever you want to train a neural network, you need to present it with what very close to that it needs to classify. So the neural network we had in the 2021 paper was a super clear, scientifically recorded recordings. So there was no noise on it, but these from the agricultural industry, there was a lot of noise. So we had to train a new neural network by simply adding more noise and doing some data augmentation and stuff. So it's like, what I'm gonna say tomorrow is that if you want to run a marathon, you need to train running marathons, and you cannot just run a 100 meter. And if you want to become good at running a 100 meter, you cannot just train marathon running. So the result is actually here from conventional. The yellow one is the really, really bad one. That's the pig screaming and being super stressed. The orange one is unhappy, but it's not like really concerning about it. You see, there's a lot of the really bad ones, except for this one, 11% really bad sounds. That's not that bad, but it turns out we did not know that when we did this. But this is actually not a Danish farm. This is a German farm. And in the documentary that you're all going to go home and watch, it actually turns out that the farmer had a really good relationship with his pigs. That he was out talking with them, and they had way more room than the law says that they are supposed to have. So it means a lot that you are actually got a good relationship and, you know, treating your animals decently in the agricultural industry. So a lot of variation between these farms, and stress level somewhere between 11, the German, the German, and 41% high stress. So for the organic ones, we actually very surprisingly see a lot of the really bad ones here in these two, right? Especially 56% of the bad calls. That's really bad. But it actually turned out a lot of variation again between 8 to 15. That this farm, Elodie was out doing a talk to the Association of Danish Agricultural Veterinarians, and the veterinarian responsible for this particular farm came down to Elodie and said, I know this farm and there is health issues there. And I think that's pretty interesting, right? That we could see it just from the vocalizations, that there was some kind of issue here, that they were not feeling well. And he said there was something about they got the wrong food so that they had back pains or something, because organic farms can move more than conventional pigs. So they need slightly different kind of foods to support this extra movement. So we know why this is really, really bad. But this was one of the things that they talked a lot about in the media in Denmark, was that there's no need to eat organic meat because they are not feeling any better because of it. But you know, we have to say these are 4 free and 2 farms. So statistically, we cannot really say anything about it. But it's just interesting. In the free range farms, basically none of the really bad calls, right? That's why I only eat bacon from free range pigs now. There's huge difference in when you see from these and then how happy these are. Yeah, there's 25 of these not so happy calls, but these calls are also the calls that they produce, like if another pig comes too close when it just found something nice in the mud. So it's not something that we are super concerned about. It's the yellow calls that is a stressed call that is associated with increase in stress hormone, increase in pulse, decrease of fitness of the animal in general. So it's definitely the lowest stress levels. And we also did some classical analysis, as we talked about, that call duration. Quite often, the shorter a call is, the more it's connected to positive valence, and the longer the vocalization is, the more negative it is. And we see a very nice, almost linear tendency here, right, between the conventional, very long calls, organic, and free range. But also the call frequency, as we talked about, there's not really that much nice tendency here, because the call frequency, as I mentioned earlier, that the higher, the bigger the chance is that it's a negative valence, and the lower, the more tendency to positive valence. So we prefer that it have a happy smile, and that's like the free range is the lowest again. Conventional is somewhere between the organic and the conventional. So the conclusion is that various levels of stress between the different types of farms. I'm trying to keep my time here. But free range pigs is the go home message, is that that's the pigs that's got the best life based on bioacoustics, but it's always also been seen in other non-bioacoustical studies. So what we want to do is that we want to improve the AI so that we can automatically pick out the different calls. So you can basically just have a small device on the wall with the farmer that then can tell you how is the situation in this stable. Is something going wrong? Are they starting to have pain or something, right? And we want to validate this tool by not only having the bioacoustics, but also including all the classical methods for assessing welfare, like stress hormones and now also facial recognition, stress detection. So thank you so much for your attention.
Thank you very much. Do we have any question?
Thank you. Hello. Thank you for the nice talk. I have a question regarding the sound production in the birds that you mentioned. One of the parameters that influences the sound is the air pressure. I wonder how about the stiffness of the muscles which control the frequencies that the birds sing. You said muscles don't contribute so much to the song, so I wanted to ask like the stiffness.
I'm sorry if I gave that impression, because the muscles are controlling the tension, right? That it's in humans there's a small muscle that's attached to a bone that connects to one of the ends of the vocal folds, and it's sort of like when you're tuning a guitar, that then you can change the frequencies by increasing and decreasing the tension of the vocal folds. And if you do the correct calculations for how precise this muscle is, it's mind-blowing. We are talking about nanometers that it takes, because people that are good at singing, not me, they can like control the frequency that they produce extremely exactly. And because birds are evolutionary bound to how good are they at singing. It's the good singer that gets the good female and can get his offspring, get his genes spread. So muscles have a huge impact on the quality of avian sound production. And that's also like in songbirds, there's 18 muscles that control the song organ. And most of them, we have no idea what they do, why there is 18, we don't know. In 2013, Daniel Düring and Koen Ellermans showed that songbirds seem to have deconnected frequency and volume control in the syrinx. So there's one muscle that controls the frequency, and then there's another muscle that controls the amplitude on top of the pressure coming from the lungs. It's very interesting.
Thank you.
Hello, and thank you for your talk. It was really cool. I have a question regarding like the emotional value, how it's correlated, the last one for the pigs, how it's correlated with the high frequency and low frequency vocalizations now. Why you choose like the pigs model, and if it's also you can apply to other species?
The pig model is basically because we are concerned about the animal welfare issues there is in the agricultural industry. And we hope that we thereby can help improve animal welfare by validating another proxy that you can use non-invasively and automatically. As I mentioned, our hope and dream is that one day the agricultural industry can use this to improve the animal welfare, and maybe also like if there's bad cases, the government, not the government, but like the state can use this. Like I think also across all of Europe, if you're driving a truck, then you have to have like a register in your truck that shows that you are actually taking the breaks that you need to do, right? So it's like if there's issues at a farm, you could have this monitor hanging on the wall monitoring bioacoustic and saying like, there's something wrong here. And then the police, animal welfare police, if that ever happens, could go in and say like, we need to improve the situation in this particular location.
Yeah, but it's something like specific to pigs, or you can also apply it like to cows. I don't know, like this.
Oh yeah, this is also something
low
that
and
we
high pitch related to the emotion.
Yeah, that's a general trend across many species with the low frequency, high frequency.
Okay.
In dogs, it can also be antagonistic if it's a low frequency, and high frequency is a friendly voice, right? Okay. So of course there will always be differences between animals, but there's a lot of animals where we see this low and high frequency. The thing is that pigs are not the perfect model species for this at all, because they can also make high frequency calls when they are very happy. So it's like,
yeah, like a squeeze of
Yeah.
happiness. Yeah.
but that's something that the neural network is not looking into the frequency, right? And you can just see like, oh, this is a call that's positive valence despite the high frequency.
Okay.
And that's why it's better than using the traditional method.
Thank you.
You're welcome.
Thank you very much. So we are running a bit over time, so I invite you to ask more questions during the break. I'm sure you will be happy to answer it, right? So, okay, thank you very much to our speaker. So time for a short coffee break, so please try to be there at 2:45. See you soon.
So I'm Elise Fouin, and I'm gonna present the results of my master's thesis on Commerson's dolphin ecology at the scale of Kerguelen Islands using passive acoustic monitoring. So the Commerson's dolphins are odontocetes. They have an odd distribution around the globe because they are found in two distinct populations. You have one in Argentina and one in Kerguelen Island. And most of the knowledge on the species ecology that we have for now is based on studies conducted on the Argentinian population. And what we know is that it's a highly coastal species, it has an opportunistic diet, and it shows some kind of seasonal distribution. And so now we want to investigate the behavior of Kerguelen's population. And for that, there is 3 chapter of research that was conducted on this population. You have one on visual observation, one with genetic analysis, and then the one that we are gonna be interested in is the passive acoustic monitoring. So the first results showed the same thing that we had on the Argentinian population. So the catalog and the visual observation showed that the species is highly coastal. It doesn't go further away than 100 meter depth, and it seems to have a resident lifestyle on the scale of the archipelago. And then the biopsies showed that there is a complex structure. Now we are gonna talk about what we could find with the passive acoustic monitoring. So the question we had was, does the species show any pattern in its distribution around the archipelago? And I'm gonna say you why this species is suitable for passive acoustic monitoring. So, as you may know, cetaceans emit sounds in their environment, both to communicate and to feed. And so the Commerson's dolphins are no exception. They emit narrowband high-frequency clicks that are at really high frequency and that we can isolate. And we can have 2 informations with these clicks. It's that we can have information on their presence and also information on their behavior, because we discriminate clicks between their intervals, and if the interval interclicks are lower than 5 microseconds, we can say that it's foraging behavior. The program was conducted using this kind of instrument, which is a specific instrument that can record clicks occurrences on a long time scale, because it has a high autonomy and it can record every timestamp for every click emitted in the environment. And it has a low detection range, around 300 meters. So it allowed us to isolate the 4 sites of the study because they don't cross. And here you have the 4 sites around the archipelago, in one specific site of the archipelago, which is the Gulf of Morbihan in Kerguelen. So for the data collected, we have a calendar on the 4 sites. As you can see, there is some discontinuity in the data because of mostly logistical issues. But overall, we have approximately 60% of recorded hours. And what we can use that information, we can use it by isolating every minute positive to detection in order to analyze their presence around the archipelago. So we have two information, as I told you. There is the percentage of minutes that are positive to detection, and then there is the percentage of minutes that are positive to detection that are positive to feeding buzz. And then we compared these results with 3 time scales. We have the interannual variability, the monthly variability to show any seasonal activity, and then the dial pattern. So first we saw that there is some kind of interannual variability. The acoustic activity is not constant around the archipelago, and it seems to decrease over the years. And also you can see that the site D is really low in detections. That's for the proportion of feeding buzzes. It seems more consistent than the DPM detections. So it would seem that the foraging behavior is a proportion that's consistent. For the months variability, we have a high seasonal pattern around the sites A and B. The site C doesn't show as strong pattern, and the site D is really few frequented. And for the proportion, you have the same pattern, like it's still consistent around the month. Then we highlighted a dial pattern, a strong dial pattern. We saw that the dolphins seems to emit mostly at night and really few clicks during the day. And for that, the proportion of feeding buzzes are following the same pattern. And to compare that with what we knew about this population, we know that it's consistent with what was observed around the Gulf of Morbihan in the archipelago, and that's it's consistent with historical observation too, because that's from a study published in 1997, and we already saw that the Commerson seemed not present in the archipelago, I mean in the Gulf, around September, so it seems that there is a high seasonality. And it seems also that they are visiting other areas of the archipelago, like the Baie du Repos, which is northern on the archipelago, and it's consistent with what the biopsy showed. And for the dial pattern, that's to be compared with another species that emits NBHF, which is the Harbour porpoise. And we don't have the fishes population data, but for the Harbour porpoise, the prey availability seems to drive the presence of the Harbour porpoise activity around their area. So we could hypothesize that it's the same pattern here with the Commerson dolphin. And so the limits are the discontinuity of the data, as you saw, and the small detection range of the instruments, because if the animal is not directly in the area of the instrument, it won't be detected, but it doesn't mean it's not in the area. And it shows only acoustic presence, and we don't know if the animal is there and silent or just not there. And for the perspectives, it would be interesting to investigate the other area where we sighted dolphins to see if they show the same acoustic activity. Thank you for your attention.
Yes. Thank you, Elise. Are there any questions on this presentation? Yes.
Thank you for your presentation. I may have missed it, if so, I'm sorry to ask the question. How did you find the clicks manually, or?
No, so the C-pods are instruments that are built by someone that also built the software to extract every clicks, and there is a classifier that is the Kerno classifier, which is able to isolate NBHF from odontocetes click from boats click. But here we have only an ABHF species, so we only kept the NBHF clicks.
All right, thank you very much.
Other questions? All right, I have one question. Are the visual and genetic studies still happening? Right
The
now?
visual one, yes, and the genetic, no.
Did they also find a decrease in presence, the same as you presented?
They didn't communicate about this. I couldn't find any information about the year interannual variability. We don't know.
Okay.
Yeah.
Okay, thank you. Do you have any idea or hypothesis as of why it could be decreasing?
Maybe they are changing their distribution around the archipelago, but I think we are missing environmental data to conclude about it.
Okay. Any other questions? All right then. Thank you, Elise.
Hi, I'm gonna make a short announcement. We are aware that the room is really hot and we're sorry about it. We are having an AC that's right here. We have fans for people that urgently need to be a bit colder, but also like be careful. If you feel bad, just tell us and take care of yourselves. And if you want a fan, we have fans, okay?
So now we will be listening to Ellie. Ellie, the floor is yours.
Hello. Is that working? Yeah. Oh, cool. Hi, everyone. So I'm Ellie, or Elisiane, and today I'm going to talk about this project that I have just started. So this is called, I think you have some other title on the program, but this is called Bioacoustics Balears. It's a new approach to spatial temporal biodiversity monitoring in the beautiful Balearic Islands. Oh, where is the chick? Oh, thank you. I'm not gonna take longer in this one. Like, why do we use Bioacoustics for scalable biodiversity monitoring? I think everyone has been talking about like why it's so great about the use of sounds for environmental monitoring, and how cool is that we have some really great recent advances in technology, and how we are processing our data much differently than we were 5-10 years ago. This is just an example of, yes. This is a study, a review that was done in 2019. I'm sure there's way more studies now if you have like a more recent review, but it's quite interesting to see that that we are seeing a lot more people using those automated recorder. This has really increased in the past 15 years or so. But when we look at which groups are we focusing on, we still have, well, there's bats here. Like I haven't seen any presentation on bats yet. Wow. But birds, of course, are the second most well-studied groups, and the invertebrates, poor guys, they are really like here on the really on the bottom of the preferences. So this is like the first thing that I wanted to highlight, that in terrestrial environments there are much more than just birds, and this is like a graph that I quickly made this morning from some data that I'm collecting for this paper that I'm preparing now. That's exactly talking about how insects are being more used in ecoacoustic studies, and how is the state of art of it so far. And it's interesting to see that while we have much more presence of or people just mentioning insects on their ecoacoustic studies, Here in blue, you have like the studies that are identifying to the species level, and it is still quite low comparing to just saying, yeah, those are insects and so what. So that's a big challenge, I think, yet, like going from bioacoustics to ecoacoustics when you talk about insects, and there's of course a lot of limitations on this, but I think we are in a very sweet spot now, like to start to talk more about insects, bioacoustics, and how to actually start to use this. And well, if you're interested, like maybe later this year, I'll be releasing this paper, hopefully. Oops. So yeah, a few months ago, when I moved to Mallorca on the Balearic Islands, the first thing that I tried to do was to find someone who was working with ecoacoustics, with bioacoustics, whatever, and there was no one. So we developed this project, me with some colleagues from there. We are developing the idea for this project that is really like to develop and implement this bioacoustic monitoring framework on the islands, with this very broad goals of identifying the soundscapes, spatial and temporal dynamics, but also focusing specifically identifying birds and orthopterans on the islands. And if you think about Mallorca, that's probably the idea that you have, and it's true. That's what we have there. I'm already homesick, but Mallorca is much more than that, and it's quite an interesting environment for this kind of studies because first it's an island, so you have like very well defined borders there. And second, you have it's not that big, it's not a huge island, but it's not like super small, so you have quite a variability of different environments and also different degrees of anthropic impact on the island. So you have like a good system to develop like a nice soundscape map of the island. This is very illustrative. Those are not my points yet, but we are working now on how to stratify our sampling for the whole island. So as I say, like I just started a couple months ago, and we are still working our way on how many sites we're going to use. We're thinking about using around 100 sites that are going to encompass like different levels of protectedness. Land use and vegetation class. We're going to collect data every season for around 2 weeks per season, and quite interesting. I'm very interested in the 24 and continuous data, in 24-hour data, as opposed to just having the morning or night or just like short clips. Because I'm also interested in acoustic niches. I think that's what everyone thought that my presentation was going to be about, but this is going to be a bit further down the road. Just quickly going through our methods. So the first thing we are doing, so we just started collecting a little bit of data, and the first thing we're doing, it's really like pre-processing. And yes, we're doing acoustic indices for that, like not the classic ACI or whatever, but we are doing some acoustic metrics to find things, for example, like clipping in the data, or even we're even thinking about using some clustering to separate sound classes and then working more detailed into them. And then we're going to move to species identification, which of course is bird nets with some manual verification. I'm having some great ideas from the presentation that I'm seeing here. Thank you so much for everyone that's working with bird nets. But with the orthopterans, it's a bit different. We are working with this package that's called Arthoptera, really to help us to more quickly having a nice database of the species on the island. And we're working with local experts as well. Our idea is really to have a good data set of taxonomic, phylogenetic, and functional diversity of birds in the island, and how does that relate with different land use and also with different seasons. Some preliminary results. So as I say, we have some nice degree of some good variation of habitats in the island, And we are trying to, for this first preliminary data that we just collected, I've just retrieved some recorders last week. So we, come on, oh yeah. So I'm gonna just present a little bit about like data that we collect on this or algahoba and almond plantations. We also have we had also record on farms, pine wood forests, and also on coastal areas that we call the garrigas. And the first thing is that's very old school. That's a false color spectrogram. And this is the colors here, they're not reflecting amplitudes. They're actually reflecting a combination of 3 different acoustic indices. And this is quite nice. This is like the 2 visualizations. It's a 24-hour, and you have like 2 different visualizations that are built with different combination of indices. Oh, wow, already 2 minutes now. And from here, just by looking at that, like we can identify a few different animal groups, or sometimes just geophony and stuff like that. With BirdNet, as I say, we just started to look into that. But the first thing you would notice is that there's a lot of variation in the number of detections, not just in the species that is detecting, but also like in the number of the detections for each one of our areas. So our idea is really to deal with this variance is really just to start to manually verifying and manually review and start to work with the confidence levels, as everyone was talking before with BirdNet. But really to separate this also into our different habitats that we have. And for the orthopterans, now we are at the stage where we are working with focal recordings and previously collected data by some local experts, and we're starting to build this nice database for the orthopterans of the island. And then we can start to move on to try to identify this from PAM recordings. Just as a curiosity, like it doesn't sound like you're gonna discover something new in there, but actually, like just working with the recordings a couple years ago, one of our collaborators found like for the first time a species in the island. So this was a species that really looked like another one, but just by acoustically he could differentiate that from the other species. So this was just 2 years ago, and we're very hopeful that we can find something interesting there. And I want to quick thank all my collaborators, the guys that are working with birds, diversity, and Jaume that is working with the orthopterans, my co-team there in the island, and my little fieldwork assistant as well. That's it. Thank you.
Thank you. Any questions?
Hello, thank you for your presentation. You mentioned in one of your slides that you were using or planning to use classifying techniques.
Sorry, the classifying techniques for?
Yes, for sounds. I don't, maybe if you can...
With the birds, you're saying?
No, before.
Before. Okay.
Before.
Okay.
Before.
Is there anything before that?
Before. Yeah, maybe it's in before. Stop.
Okay.
Clustering. Yeah, sorry, it's I didn't use. What are your aims with clustering? Because you know that biological some production by biology can be very confusing. Some species can sound absolutely different to what they are. So what is your plan here?
Well, that's a good question. So I have some experience with clustering in a very different environment. We're working in tropical environments, and we actually have some nice results with it. But really, like classifying different sound sources very broadly. But yes, we did have some nice results with that. I think that's really a first characterization of the soundscape, especially if you're gonna actually work with 100 different sites, and there's a lot of variability. But really trying to understand like where do we have more I don't know, more activity by birds, or even just like biophony, or have my biophony or geophony or stuff like that. It's really more like a first characterization, but it works quite well with the insects again. So if you want to just for birds, yes, birds are tricky. But for the orthopterans, for me, like I remember working quite well with the clustering. Like you could see like very different clusters, like with just the orthopterans. So if it's not, we don't have still have a bird net for orthopterans, so maybe that's a good way to move forward, like having at least like this first selection of the recordings that we're gonna work with.
Okay. Oh, many questions.
Thank you for your talk. It was nice. You mentioned acoustic indices. Which indices are you using?
Oh, for the visualizations.
Yeah.
Oh wow! I forgot. I can have a look for you. I know in the first I forgot because those are 6 indices. I know this red one here is like a background noise index. It's called and there's a pardon me. This is an ACI. There's also a background noise index that it's not being showed here. Yeah, this red one I'm sure is ACI, but the other ones I'll have to check to say. But who developed this? This was a group in Australia that developed these visualization techniques, and yeah, those are the standard indices they are using for it. But it's quite flexible, so this can work for marine environments as well. It's yeah.
Okay, thank you.
Just a quick question. I don't know the endemism rates at Mallorca because it's a specific situation. It's an island, etc. Do you plan to deal with this specificity of location and size of island? It can be a change the sounds that are recorded, that are used in BirdNet or etc.
Sorry, you mean like the variation in the calls used by BirdNet in the island?
Yes, BirdNet is built around databases that I think maybe there is not so many songs from Mallorca. Maybe Mallorca it's something special, and maybe
that was the first when I present the project. That was the first question of one of my collaborators, because he said that because the BirdNet was trained with data from other parts of Europe mostly, we could see some variation with the data from Mallorca. From our first rounds of reviews, like it's still looking very promising, but I think that's why we are working. We're still gonna go through a long process of manual verification, and we have a few experts that are gonna work with us on that. But that's a good question, because yeah, we might end up not having such a good results as in other areas, of course.
Thank you. And next, and for the final presentation. Yes, 1, 2, 1, 2. You hear me? Okay, so now to start. What? Yeah, louder. Okay. Now do you hear me well? Yes. So for this to start this session, Isabelle is going to present us la Société Française pour l'étude du comportement animal. So that's a great partner for first our conference, but also for many biologists, especially in France. So she's going to present us what they do. Thank you, Isabelle. The floor is yours.
Okay, can you hear me well? Yes? Okay. Actually, I prepared the slide in French, so just tell me, should I speak English or French?
If you can in English, it would be great.
Okay, I will try. But anyway, the slides are in French, so sorry for that. So good Good afternoon, everyone. I'm Isabelle George. I'm chargée de recherche CNRS at the Institut des Neurosciences Paris-Saclay. And I am here to present the French Society for the Study of Animal Behavior. So who are we and what are we doing? So the missions and the values of the SFECA are to promote the scientific study of animal behavior, to enhance the meetings and discussions between students and researchers, and to promote the diffusion of scientific knowledge of animal behavior. So the SFECA groups together all the French specialists of the scientific study of animal behavior. So it's a French society, so usually people speak French in this society, but English is okay too, of course. Our main values are animal well-being, ethic, the scientific integrity, and also equality between women and men. The SFECA, so we have I don't know how you say conseil d'administration, a council, made of 12 elected members that are elected for 3 years. We can stay no more than 2 times 3 years. And a third of this council is renewed every year. And actually, so these are the 12 members that are elected right now. And we have a bureau made of a president, a secretary, and a trésorier, the man who, checks the money. And so these people are right now Vincent Fourcassier, Vanessa Guédon, and Cécilia Houdelier. And usually we try to have people coming from different places in France, from different labs, studying behavior throughout France. So the main event for the SFECA is the annual meeting. So we organize a meeting every year. And during this meeting, of course, we give some prizes, like the best communication prize. So this is an example of the prizes that we attributed this year. And we also give other prizes. There are 4 prizes. The Prix Geoffroy Saint-Hilaire is a prize for the whole career of a senior researcher. So it is, we give it every 2 years, and it will be, the next prize will be in 2026. The last prize was given to Alain Boissy, who is studying animal welfare, and before him it was Thierry Aubin, who many of you know, I think. We also give a Young Researcher Prize every 2 years also. This year it was Mylène Dutour, who is studying interspecific communication. We also give a prize that is named Prix Parole Animale every 2 years also. And this prize is for a non-academic person who is promoting research on animal behavior for the public. And this year it was Agatha Liévard-Bazin, who wrote several books about animal behavior. She also makes some videos, broadcasts, and podcasts, things like that, to promote the scientific study of animal behavior. And since 2024, we also give a PhD prize, so the Prix de Thèse. This time it's every year, and PhD students apply directly for this prize. So if you just defended your thesis, please consider applying for this prize. And this year we gave it to Clotilde Jardat, who studied horse behavior. So we also give some grants to attend different congress. So we give 200 euros to attend a national congress in France, 400 euros for an international congress in Europe, and 650 euros to attend a congress, an international congress outside Europe. So students can apply for these grants, and this is an example of the 8 grant awardees this year. We also give some money to help organize scientific events. For example, in 2025, we help the SFECA young researchers. So I will talk about this special group a bit later. We help them organize a small event before the SFECA annual meeting. We also gave some money to help the organizers of the annual meeting. And we gave some money for you today to organize the JJBA, so the Young Bioacoustician Days. And this is why I am here today to present you the SFECA. And we also gave some money to help the organizers of the ecology and behavior meeting that will take place in Montpellier in August. So we also have what we called a label. Since 2014, the SFECA certifies that, I don't know how you say comportementalist, I mean people working with animals and helping animal owners to manage their behavior. So SFECA certifies that they know the behavior of the animals and that they have good practice in their professional activity. We also communicate, and for that we use some podcasts that are broadcasted on arteradio.com. Right now we have 17 interviews with researchers in animal behavior, and more to come soon. We also have a Facebook account, and probably we will also open an account on LinkedIn to communicate about the activities of the society. So why applying to the SFECA? It is a way to support the community and to take part in the life of society. It is a way to be a part of the network of all the specialists working on animal behavior. It is also, it gives you the possibility to apply for some grants to attend either the annual meeting of the SFECA or some other congress, always with a link with animal behavior. And it's also a way to reduce the fees to participate to the annual meeting. So the students who don't have a salary, they pay 15 euros per year. It's 25 euros for PhD students that have a salary, and it's 40 euros per year for researchers, tenured researchers or postdocs who have a salary. So you can also be on the newsletter of the SFECA. You can also receive the newsletter of the SFECA, and then you will have information about conferences, scientific meeting, grants, some job and internship offers, and you can also obtain just information about the researches of the network and the collaborations in the community. So if you want to apply and to receive this newsletter, you just have to send a message, subscribe SFECA, surname, first name, last name to this address. And I can give you the slides afterwards or the address, the email address if you want. Recently, young researchers of the SFECA decided to make a specific group of young researchers and to create a Discord to exchange between young researchers. So right now there are 344 members in this group. It is a group made for young researchers studying the animal behavior. It is made to share experience, to obtain some specific help, to build a professional network, and to meet quite often. So there are some speed meetings, some game parties, some conferences, and discussions and journal clubs. It is made mainly of master students, but also of some PhD students, postdocs, and engineers, and a few bachelor students. So if you want to join this group, you can write to sfeca.jeune.chercheurs@gmail.com. And right now, the admins are leaving the group. Some of the admins are leaving the group, so they are looking for people to administrate the Discord. So if you are interested, please contact them. And this group, they, for the first time this year, organize a half a day of discussion just before the SFECA meeting. And there were some table ronde on topics like diversity, equity, and inclusivity, animal welfare, or environmental impact of research. So thank you for your attention. Please ask me any question you would like to ask, and you can visit the website of the SFECA to have some more information.
So does anyone have a question? For example, a question? No, okay, so we will, we will be happy to have like the slides maybe later if some people will be interested to have like the email addresses, things like that to contact you. And yeah, and we would like to thank again, warmly thank the SFECA for their support, because for many of us it's the first conference, the first talk, the first meeting between young researchers, and that's partly due to the SFECA. So thank you very much. And thank you to you, Isabelle, for doing that presentation. Thank you so much.
You're welcome. Enjoy.
Bye-bye. Thank you very much. Bye.
Bye.
Hi everyone, I'm Agathe Escobar, and today I'm going to present the poster I made during my master thesis under the supervision of Barbara Klump at the University of Vienna. So this research was about inter-individual and inter-group differences in context-dependent vocalization in carrion and hooded crows. The objective was to understand if vocalization in this species can vary based on individual and social factors. So for that, we observed and recorded 13 crows divided in 3 groups of different age and sex. We've extracted over 800 vocalizations and analyzed call rate, duration, vocal duration, and vocal diversity in terms of vocal context. And we wanted to investigate the link between these variables and sex, age, dominant strength, and group size. So if you are interested in seeing more of this research and the results, you can come to this poster right there. Also, you can scan the QR code to have the PDF version, sorry, and listen to the cool calls from the crows. Thank you.
Hi, I am Samantha Aguillon and I am a postdoctoral researcher at the Chrono-Environnement Laboratory in Besançon. And I work on the Cumul project, who was born because of the context of climate emergency. The main strategy is to develop renewable infrastructures, but studies have shown that these infrastructures can have a negative impact on biodiversity and especially in flying fauna. But these infrastructures are mainly studied individually, but they are located in a landscape very fragmented and especially by roads. And so the main goal of this project is to assess the cumulative impact of renewable energy infrastructures and roads, and mainly trying to identify which species is most impacted, how the landscape context impacts the habitat use of birds and bats, and also with the main hypothesis that the noise is a key factor in the repartition in the habitat use of birds and bats in such fragmented landscape. Sorry, and this part, if you can learn more about this, you can see the poster of Marion Tiberghien, who is just here. And so all of this has will permit to develop an assessment decision support tool to simulate impacts before the installation of infrastructures, and it will be tools accessible for stakeholders. And I just stop here, and if you want to learn more about this project, you can just see my poster or talk to me.
Hi, I'm Sixue, and I come from the University of St Andrews in Scotland. And now just follow me, let's take a breath, and imagine now you are walking down a street. You meet a cat, and you say bonjour to the cat, and then the cat says bonjour back to you. What do you think? You probably think, wow, what happened? But if you meet a parrot instead, you wouldn't be so shocked, because parrot is known for copying new sounds from other species. So this ability of copying new sounds based on what an animal hears is called vocal production learning, VPL. So it's a very important trait for human spoken language. Without it, I couldn't even be giving a speech now by English. So in my study, I used pear spear nose bites as a study species because it's one of the very few mammals that have this VPL ability, just like humans and pinnipeds. Also, it has a rich repertory and highly social animals. Also, it's very small that can be bred in indoor colonies, which is suitable for our experiment on this social cause. So one of the prior studies shows that a bite can change the fundamental frequency of their social cause based on the auditory templates. So my study wants to show more evidence about VPL ability, and we used the stepwise automatic training regime. We built 8 automatic training boxes that bites can attend their vocal school in the box and show whether they can prove this ability. And we use a stepwise training regime to put the bites pay their attention to the playback rather than the operant conditioning. So if you want to see how the bite attend on their vocal school and what their course outline and what their classroom looks like, you can just see my poster. It's the third one from the front and have some cute bite pictures. And if you want to see some videos like about what the bite performance in their vocal school, you can just come and chat with me. I will show you some videos. Thank you.
Hi everybody, my name is Vittoria Turone. I'm an ethologist from the University of Turin in Italy. So my research is about Northern Rockhopper penguin. Despite being a highly vocal seabird, The only available data on the vocalization of these species is very dated. It's from 1982. So what we decided to do was to investigate a XC2 colony in South Africa. And these penguins usually live on subantarctic islands, like in the Atlantic and Indian Ocean. However, some fishermen decided to keep them as pets. But once they got to South Africa, since it's illegal to own a penguin, they just released them. And that's why they were found stranded in South African beaches. And since they can't be released into the wild, they created an XC2 colony in the Two Oceans Aquarium in Cape Town. So we aimed to provide the first acoustic characterization of the vocal repertoire of these species, and to investigate whether there were acoustic cues to individual identity in different vocal types. So if you have any questions, if you are curious about my poster, is in the other room right there, and come check it out, and thank you so much.
Hello everybody, my name is Javier, I come from Italy, and my poster is the presentation about the results of 3 months of acoustic monitoring in a wild colony of African penguins in South Africa. So if you're interested about passive acoustic monitoring for seabirds, or if you care about the conservation of this amazing penguin, come to my poster and enjoy.
So, hi everyone, for those I haven't talked to yet, I'm Axelle Duchamp, and I am here to present my master thesis that I've done in pelagies, supervised by Sky and Simmons. And the objective was to assess fine-scale behavioral responses of Harbour Seals to vessel noise by comparing activity before, during, and after noise events using AES-derived acoustic modeling. So I don't really know what else to say without spoiling everything, but I can tell you that the results aren't those you can expect. So yeah, be curious.
Hi, I'm Amelie. I'm taking you to a journey to Iceland. In this northeast, there's a fjord that's currently very remote and natural, just a few fishing vessels, but this is about to change. In a few years, there will be a huge harbor that's built to accommodate for incoming shipping from the Arctic when the sea ice is melting. And so the ecosystem will likely change quite drastically there. And so we took the opportunity to get some baseline data before this is happening. And so we had a hydrophone in the water for a year and analyzed the soundscape, and especially also the occurrence of cetaceans, of whales and dolphins, which is what I'm presenting on my poster. And we use that to inform the port before construction, so that they have to oblige to certain noise regulations, for example, but also to monitor the changes when the port is actually built. And we also used the study to test some soundscape analyses methodologies that are currently proposed as standards. So if you're interested in that, come talk to me. And what did we find? What did we hear? We heard occasionally some white-beaked dolphin whistles and echolocation clicks. This is a diel plot. If you haven't seen that before, come talk to me because it's really cool. So they seem to use that as a regular habitat. And then we also found a lot of humpback whales occasionally throughout the year, but a lot of singing in the winter, which was really cool because they usually sing in their breeding grounds in the tropic. So that was cool to hear. And then, again, this is an LTSA plot, a different type of plot, also very cool. And we heard another sound that we think is rather fish, cod or haddock, but we're not quite sure. So if you have seen this type of vocalization on a spectrogram, I'd be really curious to know.
Hi everyone, my name is Inès. I'm a master's student and I'm doing a 6-month internship in a research lab in Brest called GeoOcean. And during this internship I'm studying the underwater soundscape near Mayotte Island. So this is in the Mozambique Channel in the Indian Ocean. So I'm studying that data from from 4 years of acoustic recording. So this is Mayotte Island and the acoustic recorders that are near the island. And I've partially annotated this data set, and I'm also using AIS data to try many things. First thing is I want to try and explain the low frequency variations that have been observed on the 4 years of recording. And also I'm trying to assess the potential impacts of the global soundscapes on misty sets that are in this area. So I've done half of my internship, so this is the results of 3 months of work. So mainly about the geophony, the first approach on biophony and anthropophony. So there's a lot of work that I still have to do, and I will be glad to have your IDs. And if you want to see my poster, it's just right there. Thank you.
Hi everyone, my name is Marion and I'm doing my master internship at Odyssey, and I'm part of the Cumul project, as Samantha said previously. And so we are studying the cumulative impacts of different renewable infrastructures, such as ground-mounted solar farms or wind turbines, because we only know the individual impacts on different species, such as birds. And so I'm also focusing on noise pollution. And so my poster only show few methods to answer these different questions, because I don't have the time yet to have some results. So if you are interested, you can go see my poster on the other room, and I would be happy to discuss with you.
Hi, my name is Tifany Volle, and this is part of my PhD research. So some of you might have heard about the French debate over the pastry name called pain au chocolat or chocolatine. So the population of black redstart I studied shows geographical song variation at a small geographical scale, and they have microdialect. So males from the same hamlet will share similar songs, but songs are different between hamlets. So they seem to face kind of the same struggle as us, especially first breeding males who cannot settle in the hamlet where they sing the local dialect, but they have to settle in a foreign dialect hamlet. Ocine songs birds have different ways to learn their songs. They can have only a limited period of time to learn it, or they can learn throughout their lifetime. So what we did is follow male black redstarts by recording them throughout their life. So it lasted 8 years. And we looked if they were able to learn the new local dialect they end up in, and if they could change their song repertoire. So if you're interested, come and talk to me. My poster is in the other room.
Hi, my name is Lilou Dantin and I'm a PhD student of University of Toulon. My subject is a serious game, a digital twin, about modelization of underwater acoustic scene. And the idea is to localize cetaceans and especially sperm whales. So in the LIS laboratory, we have recordings of sperm whales in Europe, and the idea is to find where they are navigating in these waters and to avoid boat collision with them. So the main key about that is to localize them, and the classical methods has some needs that we don't always reach. So the objective of my thesis is to find a new way to localize them and to use an AI model to do that. But we don't have all the data we need to train these kind of models. So the idea is to make a digital twin. So we modelize all the acoustic scenes under the water. So the sperm whales, the trajectory of the sperm whales, and also the sound they can make and how the sounds goes under the water. And all of this would be the training set of the model. And the objective is that this wonderful model will tell us where are the whales, thanks to the recordings. And my poster is just right behind. Don't hesitate.
I'm Sarah. I'll apologize now for the wall of text. I swear my poster has more pictures on it. Basically, in short, my study was a deep dive into the striped dolphin whistle repertoire. So these guys are highly gregarious oceanic dolphin species who rely on vocal communication to maintain group cohesion. And by vocal communication, I mean whistles. So basically, I observed 58 separate categories of highly stereotyped or repeated whistle types, which I was not expecting to see in the data. So you can see here, this is a plot of 5 whistles like overlaid on top of each other, and the temporal variation is very minimal. But yeah, I just plotted that in, I think, R. Yeah, so I compared my whistles with criteria laid out by Yannick et al. For signature whistles in bottlenose dolphins. And yeah, I was kind of looking into whether or not they have the potential to be signature whistles, but I can't say for sure whether they do function as signature whistles because I don't have identity information. And there was no behavioral data adjoining the acoustic data. So yes, come see my poster. It's in the other room. Thank you.
Hello. Yeah. Hi everyone, I'm Camille Leroux, a researcher at the environmental consultancy Odyssey. And today I'll be presenting a poster on the effect of solar farm on birds, using acoustic, obviously, combined with point count survey. So this project has a specificity to focus on woodland habitat, because most of the studies on the effect of solar farm on birds have been conducted in open habitat like desert, savanna, farmlands. And currently in France there are many solar farms developing in forest. So this is a 5-year project involving multiple stakeholders. Up to now we have run into some challenges and difficulties in this project related to the recorders, the SM-Mini2, to the acoustic sampling, and also to the recorder installation, because we have to install the recorder at height, and we have to be careful that they don't fall on the solar panels and that they don't shade them. And the strong point of Envoltaic is that it encompasses 2 projects, maybe 3 in the future, to cover different type of landscape and different parts of France. So one of them is conducted by Anne-Sophie, just here, so go see her. And yeah, and we don't have results yet on this project, but I'll be happy to discuss the different steps of Envoltaic with you. Thank you.
Good afternoon, everyone. My name is Anne-Sophie Le Gal. I'm a PhD and ecologist at the consultancy West Ham in Nantes, Western France. And today I'm here to present you our poster entitled From Ears to Algorithms, Comparing Human and AI-Based Bird Detectability. So it's part of the Envoltaic 2 project, a long-term project aiming to assess the impacts of solar farms on bird communities in Western France. And this project is complementary to the Envoltaic 1 project. On this map, on the left of my slide, you can see our 25 study sites, solar farms, for Envoltaic 2, as well as the study sites of Camille Leroux for Envoltaic 1. We share the same work hypothesis. So in homogeneous habitats, the solar farms may bring structural heterogeneity, which may enhance the nesting opportunities as well as the diversity in bird species. Whereas in heterogeneous habitats, the solar farms may favor generous species and decrease overall diversity. To explore this, we are using a 2 methods approach, combining field-based expert survey with ornithologists, as well as passive acoustic monitoring with 100 meters 4. And the goal of our project is to explore the complementarities between those two methods and to implement AI into regulatory assessments. So if you are interested into long-term ecological assessment or the implementation of AI into regulatory assessment, feel free to come and have a look at our poster, and we will be happy to talk with you.
Okay, I am just noticing now that I sent the poster instead of the presentation, but bear with me. So my poster is about COVID. I think we all remember and have happy memories of COVID. Birds actually seem to have enjoyed COVID a lot, and there are many studies showing how bird abundance increased during this period. And for my poster, I tested a very simple idea, because we know noise is most prominent in low frequencies of the power spectrum. I wanted to test if bird abundance changed accordingly to the song frequency of birds. So I analyzed songs from about 50 species of North America, and I tested if this was related to another data set that I used. So it's recycling data, which is all good, from another paper that I did a collaboration with. And we actually found that, contrary to prediction, we would think that low frequency species would have increased more during COVID, but it was actually the opposite that occurred. And if you want to find out why, you come to my poster, of course, and I'll explain it.
Hello everyone, my name is Maria Paula, I'm from Colombia. I am a junior researcher at the Environmental Sound Collection of Humboldt Institute. I will advertise that repo. It's a repository that contains more than 30,000 audio from different taxa, mostly terrestrial, but if you want to explore. And Anura said is one of the projects that we worked. And I'm here to make you an invitation. It's the invitation to build together. Anura said is a project that is an open data set that we developed with many people from different countries. It is based on recordings from PAM acoustic monitoring, passive acoustic monitoring in the tropics. Tropics is a unique site that impose a unique challenge, but those challenges like overlapping many species, many sounds in a very short time of recording, also represent opportunities. So Anura set is an open data set of more than 27 hours of audio annotation from frogs. I haven't heard any presentation of frogs. I'm a frog there. We have developed some protocols, and I'm here to also foster interdisciplinary and international collaboration if you want to explore these data sets. And also, I want to tell you what are the real world challenges of develop a data set from the forest to one click. So if you want to know anything about this or about the environmental sound collection, please reach me. I'm super happy to be here.
Hello everyone, my name is Céline. I'm a PhD student based in the Netherlands, and I'm part of the Bioacoustics project. So my work is about developing deep learning models for bioacoustic, especially multitask deep learning. So just to define what multitask is, traditional models, deep learning models, are trained with one single task in mind, like BirdNet. BirdNet will output the species, and a multitask model will have more than one prediction. So for example, the species and then the distance estimation. And using multitask models instead of single task model can have some advantages. So
that I listed here. So, for example, you could combine a lot of data sets. We have a lot of data sets bioacoustics that are just open source with different annotations, and we could use them together, which is very nice. Another motivation is that with multitask model, a model will be trained with simultaneous tasks at the same time, so you will force it to be better at all those tasks together. So maybe you'll build better features for this model, more generalizable features. And that's why we did a pilot study where we compared single to multitask models for 3 bioacoustics tasks: species identification, individual identification, and vocalization type identification. So if you're curious about the results, just go by my poster. It's the orange one there. Yes?
Hello everyone, I'm Philémon Prévot, an engineer at the Elise laboratory in Toulon. And for the past years, we wanted to develop our own acoustic recorder to do mainly, like Lilou said, cetacean research, but not only. So I'm sure many of you are familiar with the requirements or challenges of good data collection for acoustics research. So some of our projects, although required us to have on-site computational abilities and some remote communication tool and tool management. So we developed the QHB Quality Life iBlue recorder. That's more of a technical poster, which has high quality, low noise acoustic recording with up to 6 microphones or hydrophones. It's a low power system, so you can set it up for month-long missions. You can run small AI models on it to have real-time inference and acoustic indices computation. So you get a view of the direct environment from your lab, and you don't need to always go on site, and that's using the GSM communication. So we've deployed those recorders on several projects across the world for the last year and a half. On long-term studies, we managed to do source localization using either one recorder with several hydrophones, microphones, or different antennas that were synchronized using GPS or atomic clock. And we used already real-time environmental analysis with AI and acoustic indices. So if you want to know more about the technical specification of our recorder, you can come see my poster in the other room. And if you want to know more about the results, you can see the poster of Lilou and Marie-Lou.
Hello everyone, I'm Elisa, an intern at CEREMA in the Environmental Acoustic Research Units, and my work focuses on the modeling underwater acoustic propagation in the Sainte Marie Channel in Madagascar. So you need to know that humpback whales depend entirely on acoustic signals to communicate and survive. And the Sainte Marie Channel is a critical breeding area for them, where communication is essential. So the problem is that maritime traffic generates noise pollution that masks their communication and can also sorry and can cause collision risks. So the goal of my internships is to model this acoustic propagation to better understand the impact of cheap noise on these marine mammals. So if you want to know more about it, my poster is just here. So thank you.
Hello everyone, I'm Bianca from Conisma in Italy, and this is the first and preliminary study about the characterization of the whistle emission of Tursiope truncatus.
in the Northern Union Sea. The aim of the study was to investigate the acoustic behavior of bottlenose dolphin, focusing on the identification of signature whistles, which are individual, distinctive, frequency modulated sound. We did the identification using the CGIT criterion, according to which a whistle category is considered the signature whistle type if during the visual analysis there is a sequence of at least 4 whistles, and if at least once the 75% or more of the whistles occur with an inter-whistle interval of 1/10 seconds. And here below you can see an example of signature whistle classification. Using Pratt, we found 6 different categories of signature whistles, and using PalmGuard we extract the pitch contour of the fundamental frequency. And on these 10 acoustic parameters we did the statistical analysis. Specifically we did a linear discriminant analysis to evaluate the correct classification of signature whistles in the different categories using only the acoustic parameters. And then we did the principal component analysis to evaluate the most correlated parameters with the first 2 principal components. If you have any questions, my poster is over there, and I will be happy to answer you. Thank you.
So, hello everyone, my name is Arthur Touchet, and I will present the results of my internship that I did at the Inès, so the Équipe of Neuroethologie Sensorielle in Saint-Étienne. So, I studied the vocal production during approaches in chimpanzees. As you know, signaling involves transferring of information and of intentions, and it's especially important during uncertain contexts. And approaching individuals involves a degree of uncertainty both for the approacher and for the approached individual, as the outcome of the approach may depend on social relationships as well as on the overall context. So what we did was we studied 2 groups of chimpanzees, of captive chimpanzees in a zoo, and we recorded their behaviors and also their vocalizations, of course, during the approaches. And one of the main results that we had is that the approacher was most likely to come into contact with the approached individual if they had vocalized. So we already see that there is an impact of the production of vocalization on the approach. And we are doing some more exploratory analysis at the moment because this is still an ongoing study, but we suspect that the type of calls during the approach could have an effect on the outcome of the approach, and also the social environment and a lot of other factors that we will explore. If you have any other questions or want to know more about the study, you can come to see my poster. This is the one that fell during your presentation earlier. Sorry about that. And yeah, you can come to see me if you have any questions. Thanks.
Hi, I'm Marie Buisson. I work at the University of Toulon. And so the idea of this study is to try to estimate cetacean distances using passive acoustic and as such cetacean trajectory. So fieldwork were conducted in the Mediterranean Sea, and sperm whales were recorded with a compact and near surface antenna composed of 5 different hydrophones. You have the picture just here. And so in the recording there is some information about the distances. We have the beginning of the click and the arrival of the surface echo, and thanks to that we can have the time difference due to surface echo for each hydrophone. And moreover, we have the time difference of arrival between each pair of hydrophone, and thanks to that we can readily estimate sperm whale direction, so azimuth and elevation. Despite all this observable, estimate distances remains very, very challenging in case of compact antenna. So the idea of this poster is to try to combining the information included all this observable thanks to a deep learning model to try to estimate distances. So we give to our model the delay due to surface echo, the time difference of arrival and the direction on arrival to try to obtain the distances. And so firstly, this method was validated in case of spaced hydrophone, and after that we obtained the depth profile in case of the compact antenna dataset. So if you want to know a little bit more about this result, my poster is the other one. Thank you.
Oh, it good. Okay, everything has
been moved around, so enjoy this very messy slide. Anyways, my name is Hannah, and I did my master thesis on vocal individuality in carrion and hooded crow nestlings and fledglings. So in order for an individual to be able to recognize another individual by its voice, there need to be like distinct acoustic features in a call, and this is known as vocal individuality. And we know from studies on other bird species that even very early on in ontogeny, this vocal individuality can be present. And we also know that there can be a change in how individualized calls are with age, depending on the adaptive value of being recognizable by voice. But it is still kind of unknown what factors exactly influence such changes. So we really need more studies on different species, ideally of different or varying life histories. So therefore I looked at this in carrion and hooded crow hybrids. And for this we hand raised and recorded them in the nestling and the fledgling stage to answer the following research questions. So firstly, I wanted to know whether vocal individuality is present early on in these birds. Then I also wanted to know whether this individuality or the degree of their individuality differs throughout different age classes. And also I wanted to see if it also is different between behavioral contexts, because this can also influence the individuality of calls. So if you want to know what the answers to these questions are, check out my poster. It's like right next to the entrance above the trash can. So let's hope it doesn't fall, and I'm happy to talk to you if you are interested.